Okay, hi everyone. So today I'm going to be talking about the prediction section from EA3. The prediction section will serve two really nice purposes for all of you today. The first one is that it'll serve as a sort of an important proof of concept for some of the concepts that we've been talking about during the workshop, especially surrounding polygenic prediction and polygenic scores. And second, since we'll soon be finished with EA3 and these scores will be available to you, they can give you a preview for some of the things you can think about doing in your own research. Okay, so these are the main collaborators on the EA3 project, in case you're interested in who all of us are. The names that are highlighted in bold are the individuals who have been particularly involved in the prediction section of EA3. Okay, so the scores that I'll present on today are constructed using LD-Pred, which you all should be very familiar with by this point, both from the theoretical aspect and because you've actually constructed an LD-Pred polygenic score using the EA2 meta-analysis. And the two prediction samples that I'll be presenting on are both AdHealth, which again you're very familiar with, and also the health and retirement study. This is another large longitudinal US representative study that a lot of polygenic prediction analyses are done in. And Isu mentioned this point earlier in the week, but in this prediction section, we're actually interested in making comparisons between our scores in AdHealth and in HRS. So we'll impute both of our data sets. So this is a little bit different than what you've done. You were using genotyped SNPs. Here we'll be imputing both data sets, and we'll be using HapMap3 SNPs from those imputations. This allows us to make fair comparisons, and also we know, as we talked about earlier, that those SNPs are known to be reliably well imputed. A few important descriptives that allow us to differentiate between the AdHealth and the HRS samples. So in AdHealth, we have a higher mean years of education completed for the AdHealth respondents in comparison to the HRS respondents. This is in line with modern demographic trends in educational attainment, where younger people are now completing more years of education than our older individuals. So you can see a higher mean birth year in AdHealth than in HRS. Okay, so you've also done this in the problem set, but I wanted to just walk through it because it's sort of the key to the results that I'll present in the prediction section. So our measure of predictive power is called the incremental R squared, and so I'll use those two terms interchangeably here. We get it with just a simple two-step process. So in the first step, we regress our phenotype of interest, which will often be educational attainment, on our control set of variables without our polygenic score included. And then in the second step, we'll rerun the same regression, but this time we'll include the polygenic score. So for quantitative phenotypes like educational attainment, the measure of predictive power will be this incremental R squared, or the change in R squared from the augmented regression. Sometimes I'll be talking about binary phenotypes. It's exactly the same process, except we'll be calculating the change in the pseudo R squared. And finally, all of the 95% confidence intervals that I'll present here will be bootstrapped with 1,000 repetitions each. Okay, so you've seen this figure already, but this is the current meta-analysis of EA3. We have about 770,000 individuals in the meta-analysis, and we've currently identified 529 genome-wide significant independent loci. Just a reminder that soon the UK Biobank, the second release of the UK Biobank will come out, and our sample size will be over 1 million individuals. Okay. So currently in HRS and AdHealth, the predictive power of the polygenic score for predicting educational attainment in HRS is about 9%, and you've seen these numbers, I think, already, but in AdHealth we're getting close to 11%. So this is really exciting for us. This makes us some of the highest polygenic prediction numbers that we've seen for any phenotype, and particularly for social science phenotypes that a lot of us are interested in. This graph on the left shows the important relationship between increasing sample size and increasing predictive power of a polygenic score, like we've talked about. So here, the dots represent the different sample sizes for the EA meta-analyses over time, starting back in 2013 with EA1 and going all the way up to the current meta-analysis that we're looking at today. Mm-hmm.
Do you have like an intuition as to why it's more predictive in AdHealth than HRS?
So Megan, I think Megan's going to highlight some of those in the cross-cohort prediction section, so we'll leave that for her. But yeah, we have some ideas about what characteristics might be driving those differences.
Are they significantly different from one another?
Let's see. I don't think, I think they are. They probably are. We can pull up the tables in the Q&A if you want to look at them. Yeah. Okay. Okay. Yeah. Mm-hmm.
Can you talk a little bit about the bootstrapping, how you do this?
So
Unless you want us to just talk about it in the coffee break.
we can talk about it in the coffee break. That's fine. Yeah. I can show you the code for how to do it too.
Similar. Do you use? We can just talk about this later. But do you use the survey weights, or how do you?
We don't. I don't think we use the survey weights in here. No, we don't use the survey weights here. Yeah.
This is just individuals, and you interpret this as like individuals in the sample. How do you do it there?
Mm-hmm. Yeah. Okay. So I'm going to use some notes here because I'll forget what I'm talking about if I don't. Okay, so we're also interested in other phenotypes that are related to educational attainment or related to cognition in EA3. So we'll begin here with educational phenotypes that are available to us in both the HRS and the AdHealth dataset. So here we considered 3 indicator variables related to educational attainment. The first one was high school completion, the second one was college completion, and the third one was grade retention, which is an indicator variable for whether a respondent had ever repeated a grade or not. Okay, so the phenotypes are on the x-axis, and on the y-axis they're plotted against this time the pseudo R-squared value, again a measure of predictive power. Okay, so in both of our prediction cohorts for all 3 of our phenotypes, the PGS is significantly associated with our 3 outcomes. And so for ease of visualization here, at the bottom of each bar, I've plotted the incremental pseudo R-squared value for each of the phenotypes. So for AdHealth, which is in the blue bars, the predictive power is about 6.2% for high school completion, is about 8.2% for college completion, and is about 4% for grade retention. And the comparable values for HRS are there in the gold bars. And I guess for more of a substantively meaningful interpretation of the results, we also calculate the marginal effect for the polygenic score for each of our variables. So for AdHealth, a 1 standard deviation increase in the educational attainment PGS is associated with a 4.2 percentage point increase in the probability of completing high school, all the way up to a 14.8 percentage point increase in the probability of completing college, and a 7.1% reduction in the probability of repeating a grade.
Robbie, are these just the SNPs, or is that the entire polygenic?
We include all SNPs in the polygenic score. Yep. So it. What?
Is it all SNPs, or were they pruned somehow?
No.
With the LD pred?
So it uses LD-pred. It's all SNPs using LD-pred. So it's exactly like the score that you made, except instead of just 1 chromosome, it's all of them. We're using it.
3 SNPs. So I guess we cut, but we don't prune for LD. We just kind of prune to get a common set of SNPs.
Yeah.
You could think of some SNPs being like weighted down very heavily, right?
Yeah.
Like to 0 or
Yeah. I think so. Yeah. Mm-hmm.
So you impute first, and then you prune to a common data set where you cut things that are not imputed well, or?
So we're going to pull out the SNPs from the imputed data that are, or the HapMap3 SNPs from the imputed data. We'll just subset by the imputed data.
Subsetting to the HapMap first.
Yeah.
Okay. Got it.
Yeah. Okay.
Sorry, if you just drop anyone who is not of European ancestry, you include people who
Yes. Yep. These are.
responded more than one.
Yeah. So these are only Europeans that we're presenting on. Yep.
Wait, I'm sorry. Pause my question. What is the advantage of subsetting to the HapMap3 sets?
So there are 2 advantages here. The one is that they're reliably well imputed, so that's good. And the second one is that we want to make more direct comparisons since we're using 2 data sets here. So we want to be able to compare to a common set of SNPs. Yeah. Mm-hmm.
Why focus on the change in the R-square instead of, like, let's say, actually trying to predict? I mean, usually, like, let's say when you're doing data mining, you find a model and you're trying to predict out of sample. What you're trying to do is really try to figure out if your prediction of you actually went to college or not went to college and what share of people you actually correctly predict, right?
So I don't understand the question.
I mean, this is a measure of how well your fit or how much your fit improves, right?
Mm-hmm.
But maybe, let's say, if I took the model and I try to figure out what the implied probability for you of going to college is. Let's say the predicted probability is larger, greater than, equal to 1/2. I say you went to college. Usually, like in these type of models, you would use that to see how much you actually do predict correctly as a way of testing how well your model is.
So you're talking about like area under the curve type measures.
I'm not sure if it's an area under the curve measure. It's right, because let's say I'm just flipping a coin and trying to figure out if it's biased or not, and then I have a model that says like I should get mostly heads. Then I look at something and I would try to predict if my model is saying that you should only see, I don't know, 10 heads. And I look at the right?
Yeah. So I mean, I think that if you're just looking at binary measures, then there's a lot of measures that do this kind of thing that you're talking about. And I mean, I guess in this case, these are all binary measures. So you could do that here. If you were trying to predict a continuous measure, it's more tricky to think about what.
No, that's why I'm asking here, because in a sense, I can understand why in a continuous measure, and even there it's not really continuous, like right. But here it seems like you have other measures which you may be able to give us a better picture of how much.
Yeah. I mean, if we want to compare across both continuous and binary outcomes, though, R-square is defined for all of those, and so I mean, like, it's true. You could do that.
No, but you can overcome certain things. Right? Imagine that just my principal components already predict me going to college, so the extra, like the GWAS information, may not be giving us this additional. It improves our fit, but it doesn't really improve our prediction in a sense. That's why I was asking if you.
I think
have
both
you
measures
noticed?
are interesting. I think this is basically the most conservative measure that says this is the genetics contribution. But if you want to write down a model that uses that and other things, that's also possible. So both are relevant. This is just the more conservative measure. This tells you about the genetics. That's what this is. Or am I not understanding the question? If you want to predict, we can do other things.
We can talk about it.
Yeah. I mean,
We could do those analyses. Yeah. Okay. Okay, so I just flashed up the comparable values for HRS there. Sort of the takeaway from these analyses is that the polygenic score seems to be explaining a modest amount of the variation in variables that we think capture important transitions in sort of the American education system, right? Since these are both American-based data sets, or US-based data sets. Okay, here's sort of another interesting way that we wanted to depict our results. So here we've divided our polygenic score into just 5 different quintiles. So that quintile 1 here on the left indicates low polygenic scores, quintile 5 indicates high polygenic scores. And then what we've done on the y-axis is we've simply graphed the mean prevalence of college completion in each of our quintiles for both of our data sets. So first, as we would expect, mean college completion in HRS, since we're using older individuals, is lower than in HRS in each of the data sets. But we also see sort of this nice linear relationship that we expect emerge between quintile and mean prevalence of college completion. So in particular, we find the difference between the first and the fifth quintile in ad health quite striking, with about a 43% difference in mean college completion between the first quintile and the fifth quintile. And there are the comparable estimates for HRS as well. So another sort of nice descriptive way to look at the results. Okay, so we're also interested in educational achievement and cognitive phenotypes that are unique in both data sets, so that we can't directly compare in both of our data sets. So I won't go into all of the details of each of the measures that we look at, but of course, if you're interested in those, or you're interested in related variables in HRS or ad health, I'm happy to talk about those in the Q&A or after the session. So I'll just go through the results now. So in ad health, we have a measure of verbal cognition. That's sort of an augmented version of something called the Peabody Picture Vocabulary Test. And also in wave 3 of ad health, researchers collected the transcripts from the high schools of ad health participants, and they calculated using those transcripts both overall and subject-specific GPAs. So that's a really nice measure we have in HRS, or in ad health. The blue bars here, which represent ad health, show the results of the predictive power. So for Peabody, we have a predictive power of our polygenic score of about 6%. And then we have about 8% for overall GPA and 6 to 7% for those subject-specific GPAs. So some measures of academic achievement. And then in HRS, which we have depicted by the yellow bars, we have measures of total cognition and overall cognition. And also we were interested in measuring some wave-to-wave changes in these total cognition and verbal cognition variables. And finally, in waves 10 and 11 of HRS, there's a measure that's equal to 1 for respondents who report ever being doctor diagnosed with Alzheimer's and 0 otherwise. So we were interested in all of these cognition phenotypes in the HRS data set. We see that the predictive power of the score is about 2.25% for total cognition and about 4% for verbal cognition, but we didn't find any significant associations with our wave-to-wave change measures or with Alzheimer's. So this is a graph that contains a lot of information, but sort of the takeaway here is that the score seems to also explain a modest amount of the variation in some very crude measures of cognition and academic achievement. Okay. This is another interesting supplemental analysis that we did in the prediction section. So here we've created for each data set 22 individual scores, 1 for each chromosome. And on the x-axis, we're plotting the chromosome length. On the y-axis, the incremental r-squared of the individual by chromosome polygenic scores. And so as we would expect, we see a nice linear relationship emerge between chromosome length and the predictive power of the bi-chromosome polygenic score. And we think it's really interesting that with these very large sample sizes that we have in EA3, if we just add up the predictive power of some of the longest chromosomes, we're now out-competing the original educational attainment polygenic score back from EA1 in 2013. So we've made a lot of improvements. Mm-hmm.
How does this compare with heritability, heritability explained by each chromosome? Is it fairly similar?
Oh, I don't know. I guess we should do that. I don't know what the bi-chromosome heritability is. Yeah. Mm-hmm.
Sorry, is the intuition here just that there's more information?
Yeah, exactly. Yeah, it's sort of like a nice sanity check. It's also nice to see very similar results for both of the data sets. Okay. So in sort of a last set of analyses, we wanted to benchmark the predictive power of our polygenic score against some other variables that have historically explained a lot of the variation in educational attainment. So a reminder that we'll start here with the predictive power of the educational attainment score at about 11% in AdHealth and 9% in HRS. And then we've picked out some common socio-demographic variables that are often available in some of these data sets like HRS or AdHealth. And so we've also calculated the predictive power in a similar way as we do the polygenic score for all of these variables. And so you can see here that with the large sample sizes that we have in EA3, we're now beginning to outcompete some variables that have historically explained a lot of the variation in educational attainment, like household income. And also, even though the polygenic score is underperforming variables like mother's education, father's education, or verbal cognition, the predictive power is now so high that we're beginning to compete with some of these variables as well. Mm-hmm.
Clarifying question. So this exercise is not that you're adding on top of your PGS
right.
also this, but you take PGS out and you do mother's education.
Right. So in this column, what we do is we take whatever variable this is. So we take educational attainment and we regress it on our standard set of controls, right? Without any of these variables. And then for each variable, we add it back in and calculate the change in R squared, right? So it's the predictive power of that variable.
Mother's and father's education, how's that coded? Is that the highest?
So it's coded in, I believe, let me remember this. In AdHealth and HRS, it's coded exactly the same way as we code educational attainment for the prediction section. So these are all sort of just continuous variables for education.
It means that the one they call mothers and fathers, is that just an average or?
It's also a continuous variable for completed years of education.
How is it constructed?
They just ask.
You sum mother and father. Is that what you do?
Oh, this one?
Yeah.
Yeah, we just sum mother and father. Yeah, you were asking about this variable. Oh, sorry. Yes. Yep.
Oh, wait, or is it the R squared from just the regression with two controls?
Yeah, it's the two controls. Yep. Does that make sense? Yeah. Mm-hmm.
Do you have a sense of like, so we'd expect mother's education, father's education to have some sort of, like, there would be some collinearity between the PGS and those. Like, do either of those attenuate when you include the PGS?
So, does the predictive power of the score attenuate when you add these Yonis controls?
The other way around. So I guess mother's education, we would expect something about child PGS to be in there, right? So my question is, like, does the predictive power of that attenuate if you include PGS?
I assume it would. We should do the we haven't done the analysis, but I assume that it would. Yeah. Yeah. Mm-hmm.
So I see you guys are controlling the birth year by sex in the model.
Mm-hmm.
Is that usually the case?
Yeah, that is. It is. It's usually the case to include that as a control. Yep. Okay. So in this last analysis, we were also interested in what happens to the predictive power of our polygenic score as we throw in a bunch of controls. And so what we do here is on the left-hand side, we run the regression of educational attainment on whatever set of controls are here. And then we add in our polygenic score and calculate the R squared again. And so you can see here that we begin to see some attenuation when we start including variables as we would expect, like household income, mother's education, father's education. But even after we throw the whole list of controls that we consider here at the regressions, we still have a relatively high incremental R squared of our polygenic score of about 4% for both data sets. And so, you know, if you've been reviewing the GWAS literature at all, these are still pretty high incremental R squareds, even after sort of this very extreme test of throwing all of the controls at the model. So this, of course, might speak to the ability of us to use highly powered polygenic scores, polygenic scores created from highly powered GWAS as controls in some of these data sets going forward. So the conclusions are sort of broad but exciting. First, the scores from EA3 are among the strongest polygenic predictors that have been constructed to date. And of course, as larger samples are increasingly available to estimate weights for polygenic scores, their predictive power rises. And soon EA3 will have one of the first scores constructed using over 1 million individuals. And also, you know, in the SSGAC, we really like this idea of hopefully potentially being able to use polygenic scores as control variables one day, which I think we have some evidence for here. And that's it. Thank you.